Okay, everyone, welcome back. I hope you're feeling re-energized after the long poster session. And it's my great pleasure to introduce J Paton. So Joe is a group leader and the director of the neuroscience program here at the Champalimo in Lisbon. And today he's going to tell us about time, control, and the nervous system.
Thanks, Rui. It's Paton, by the way. Sorry for the delay. Age happens. So if I want to see what's on my screen down here, then I need my glasses. So I'm going to do something a little different today than I normally do in a talk, which is to start by presenting a bunch of ideas that both frame the work that I will tell you about later and also perhaps provoke you a bit and maybe, in however small a way, changing the way you think about the brain. Now, before diving in, I should say that if these ideas intrigue you, you can read a deeper treatment of them in a recent review that Carolina Heimroll, Felipe Rodriguez, and I wrote, the reference to which is here at the bottom. Okay, so the first provocation is that I will assert that time is the single most important concept that one needs to consider when studying not only the nervous system, but in fact all biological systems. And that's because all biological systems are fundamentally control systems. And controllers, of course, they regulate the state of some target dynamical system by sensing features of that system, passing those observations through some control law, which determines how the controller acts to keep the regulated system within some set of target conditions. Of course, dynamical systems, by definition, exhibit lawful structure in how they evolve through time. And you can improve your controller by building knowledge of the dynamics into your control law. Take, for example, the thermostat of this building. If it knows that the building will heat up and cool down periodically with day-night cycles, it can use that knowledge to minimize error to maintain a more stable building temperature. Now, in fact, this isn't just useful, but it's actually been proven by Conant and Ashby in this paper, sometimes referred to as the good regulator theorem, that optimal controllers must absorb structure from the system they regulate. And I quote, "In this paper, a theorem is presented which shows, under very broad conditions, that any regulator that is maximally both successful and simple must be isomorphic with the system being regulated. Making a model is thus necessary." Now, I want you to hold this thought right here, that making a model is necessary, because I don't think it's quite right, not at least in the way we commonly use the word model. But I'll come back to that in a moment. Nonetheless, I do think Conant and Ashby were onto something profound in pointing out that optimal controllers must absorb some manner of structure from their target systems. And again, because these are dynamical systems, the structure in need of absorption is fundamentally temporal in nature. And one reason control is so improved by this process is that it enables a switch from feedback control to one that involves anticipation. And I want to provide a more concrete intuition of how this works by going back a couple of billion years, before nervous systems even existed, to this prokaryotic cell floating around in primordial seas. It too was faced with control needs, specifically in this case to control its osmotic balance and overall cell integrity. Now, this is a bit of an evolutionary just so story, but the idea is that there were existing membrane repair mechanisms that were triggered by the rising calcium levels that accompanied membrane damage. Calcium would rush into the cell. It would bind to a matrix of actomyosin lining the cell wall, creating a contractile ring that would bring the edges of the hull of the membrane closer together. Second, calcium would bind to molecules that later got co-opted for synaptic release machinery that mediate exocytosis of internal vesicles, adding new membrane to help patch the hole. So these are purely feedback control processes. However, mechanical stress on the membrane reliably preceded the hole in the membrane. And so the idea is that this created selective pressure for some kind of mechanism that could translate that reliable feature in the dynamics of the target system into preemptive action. And here's the just so story. The mechanosensitive calcium channels evolved to do just that, coupling mechanical stress in the membrane to calcium influx, triggering contraction of the actomyosin and vesicle release, maintaining cell shape pushing off of whatever was leading to the mechanical stress and thus preventing membrane rupture altogether through control mechanisms that are anticipatory. Now, I like this example because it highlights how biological intelligence is most often fundamentally in the world. It's constructed iteratively out of time over physical stuff, and how much of its cleverness is achieved not through explicit knowledge. The cell here doesn't have an explicit model of the world where it knows that membrane stress leads to damage. Instead, its knowledge is, as the philosopher Michael Polanyi would say, tacit and implicit. But my read is that many people have interpreted Cosyne and Nashby's assertion about the necessity of model construction for optimal control as meaning that explicit models are needed. But this is clearly not the case. And I think it actually saves the theory to allow for this more implicit form of, I wouldn't call it a model, but absorbing structure from the system you regulate. Now, if we zoom ahead a couple of billion years or so to look at vertebrate animals, we now have complex nervous systems, but we still see signs of how absorbing system dynamics can lead to anticipatory control, even in relatively hardwired circuits. Take, for example, muscle spindles. These are fascinating sensory organs, about 30,000 of which are distributed throughout your 600 some odd muscles. These little guys support a lot of complex function, much of which, frankly, we still don't really understand. But here I want to focus on one function they support, which is the short latency stretch reflex. So this reflex is mediated by sensory endings in these intrafusal muscle fibers that convey different features of the state of the muscle monosynaptically to alpha motor neurons to activate the skeletal muscles that move the body. Now let's consider a stretch reflex that couples static muscle length to muscle activation through some function that looks something like this. Because we're coupling current muscle state to action, this constitutes a form of reactive or feedback control. Turns out there are a variety of different muscle spindles across vertebrates, and the spindles of tortoises and turtles create a stretch reflex that looks more like this. It's kind of interesting considering how slowly they move. But if we add in some combination of contribution of the derivative of muscle length, or its velocity, this can be seen as a proxy for where muscle length will lie along this static curve in the near future, thus creating a stretch reflex that includes an anticipatory component. Again, perhaps not coincidentally, this is what the stretch reflex of more agile vertebrates, like ourselves, actually looks like. Once again, we see here how reproducible dynamics in the system, the fact that muscle velocity predicts future muscle state, has been absorbed into the properties of a controller for anticipatory regulation. And again, no explicit model here of the environment. And of course, biological systems, and brains in particular, possess layer upon layer of these kinds of mechanisms. And those two examples I just went through represent cases where very stable and widespread control-relevant dynamics get absorbed into relatively hardwired control mechanisms via evolution acting on the genome, ultimately. And we can see large classes of similar cases in the vertebrate brain, for example, in the spinal cord and brainstem, where reflexes, muscle synergies, and central pattern generators are good exemplars of relatively automatized forms of control that nonetheless reflect good regulation. But what if the control-relevant dynamics you want to absorb are more novel? The environment might have changed in recent years. Or is idiosyncratic from the perspective of a species. If I'm a city rat or a country rat, I'm going to be exposed to very different environments and engage in very different patterns of behavior. Well, if the brain is to keep following this prescription for being a good regulator, it needs to absorb idiosyncratic control-relevant dynamics within the lifetime of the organism, that is, through learning. And for that, in the vertebrate brain, we have this collection of brain systems positioned more anterior along the neuraxis, and I'll just single out cortex, the basal ganglia, and the cerebellum as three big ones where we believe a large part of their functionality rests on an ability to learn from experience. But why multiple learning systems? Each possessing its own stereotyped circuit architecture that would seem to suggest a distinct computation. Whoops. You know, this basic nervous system plan has been in place for order 400 million years, and we're still pretty far from agreeing on what the kind of formal contributions of these different brain systems is to the overall functioning of the brain. We suffer, I think, from a bit of an aperture problem, whereby focusing on specific problems or circuits to gain tractability, and that's of course understandable, we're actually biased away from thinking about brain function in a more integrative manner. In the rest of the talk, I'm going to present some work reflecting on our early attempts to help address that issue by jointly studying multiple brain systems. But just to wrap up this first part, I hope you've gotten a sense of why I stated at the outset that time is so central to all, not only nervous system function, but biological function. It has its roots in the normative view that good regulators of a system must absorb structure from that system, and that the relevant structure is fundamentally temporal. And this is deeply related to ideas that Claude Shannon was getting at in laying out the basis for information theory. As he says here, the main point at issue, when talking about good codes, is the effect of statistical knowledge about the source in reducing the required capacity of the channel by the use of proper encoding of the information. In fact, the title of this talk is a bit of shameless theft from Norbert Wiener in this paper, in which he folds together control and communication, emphasizing the central importance of time when you do so. And a lot of what Wiener wrote here is a less crisply formalized pointer to Conant and Ashby's work 20 years later. So if you want to get perhaps a more eloquent and deeper description of the concepts I've been trying to get across for the last 10 minutes, I would suggest you go read these 3 papers and let their messages stew in your mind for a while. Now, one additional point that we've tried to make is that both evolution and learning can be seen as having this same objective. With evolution shouldering the burden when the relevant information is very stable over time and widespread, and learning coming in when it is novel or idiosyncratic from the perspective of the species. The point is that no matter what, time is always there. It isn't the sensory modality that we can sense its passage, it's the informational fabric into which all other features are woven. Okay, so for the rest of the talk, proselytizing complete, I'm going to focus on some work, much of which is still in progress, that we've been doing to try and tease apart the respective contributions of 2 of these learning systems, the basal ganglia and the cerebellum, which we view as systems for learning programs or policies for behavioral control that operate on categorically distinct control spaces. And this work is largely being performed by a postdoc student team of Teresa Serrades-Duarte, Simone Zamora, and Hugo Rafael, with important contributions from Megan Carey, Coralie Herant, and John Krakauer. And apologies to all you cortical chauvinists out there. I'm not going to talk about cortex today, but suffice it to say, we view it as a representation learning system. However, Caroline Eimerl will give a talk on Tuesday morning at the agent-based workshop about exciting work she's been leading on the topic of how predicting actions and not just states drives learning of representations with very useful properties. Again, long-term, this is aligned with our view that prediction is important only insofar as it improves control. Okay, so you might have picked up on the fact, due to some of the language I was using, that we frame a lot of our thinking about adaptive behavior through the lens of reinforcement learning, because we view it as a really accommodating agent-level framework for thinking about adaptive behavior. And I want to point out first that reinforcement learning is defined not by characterizing learning algorithms, but by characterizing a learning problem. And that problem is how do agents, artificial or animal, learn how to behave through interaction with the environment. And in the RL framework, the objective of that learning is to learn policies or programs for behavioral control that do something like maximize expected cumulative reward. Now, of course, the field of reinforcement learning provides an ever-expanding set of algorithms by which that problem might be solved, as you can see here in this schematic from OpenAI. And in the lab, we often look to these algorithms not for the exact description of what different neural circuits might be doing, but rather for inspiration when we try to construct models of the processes that neural signal circuits seem to be implementing, taking into account the constraints that we see in the biology or animal behavior. And I'm not going to talk about this today, but here are 2 relatively recently published studies from the lab where we engage in trying to build these more neurobiologically constrained reinforcement learning models. This focus on reinforcement learning has led us to spend a good amount of time studying the basal ganglia, because it seems to play a central role in learning policies for behavioral control. And the general idea goes as follows. Information from an extremely broad range of functional territories in cortex, almost the entire cortical mantle, as well as the thalamus, converges in the major, provides the basal ganglia with information about generally the situation or the state of the world an animal finds itself in, as well as the actions that are on offer there. And the state and action-related information converges in the major input area of the basal ganglia with extremely dense input from midbrain dopamine neurons that are thought to carry an important teaching signal called a reward prediction error. And it's the convergence of this teaching signal with the state and action-related information that's thought to produce plasticity in the striatum that allows it to learn something about the value of available actions in that context. And then in turn, this action value related information can be used to derive policies for behavioral control, which can then influence the selection of actions through the various extrinsic connections of the basal ganglia with downstream motor structures, either in the brainstem or indirectly in cortex. Now, as I hope I made clear in the introduction to this talk, in order to perform such a function, this system needs access to some kind of temporal basis for absorbing dynamics involving observations, actions, and rewards. And so to get a sense of what form that temporal basis might take, we've been studying behaviors that isolate and exaggerate the use of temporal information. And one of those behaviors is a time-based decision-making task that goes as follows. Animals, we study rats and mice, are placed in a behavioral box where there are 3 nose ports positioned along a wall. They learn to initiate trials by inserting their snout into a central port. This elicits a brief auditory tone followed by a variable time delay of silence before a second tone is played. And at this point, animals are free to move to the left or the right port to get reward, depending on whether they believe the delay between those 2 tones was shorter or longer than a learned category boundary. So animals are making these categorical judgments about the duration of silence between 2 stimuli around a learned category boundary of 1.5 seconds. And one interesting thing about this task is at the time of the second tone, there's nothing explicit in the environment that's telling the animal where to go to get reward. So instead, they have to rely on some kind of latent, dynamic, evolving representation that's somehow related to elapsed time if they want to get rewards. And I think this capability, or this capacity to operate on kind of latent, dynamic, internal variables is sort of a hallmark of what we might call a cognitive process. So there may be kind of general lessons from studying this task about cognition. Okay, so animals can learn to perform this task quite well. Here's an example of a psychometric curve. The probability that the animal makes a long choice is a function of the different intervals we present to them. And we've used this task over the years to ask a number of questions related to topics ranging from action suppression and motor control, the role of neuromodulation and temporal estimation, to probing for signatures of efficient coding in the cognitive variables that animals use to solve the task. But one of the first questions we asked was if the basal ganglia are involved in learning policies that map temporal information to left and right movements in this task, what neural signals is it using to do so? So I'm going to compress a lot of different analyses into one slide, but the basic take-home story is that if you record from striatal populations during this task, you find what you might find in many areas of the brain across many different tasks, namely that neurons are active at different times, tiling the entire delay period when we're asking animals to keep track of elapsed time. And this is what you need to create a clock, right? Because it creates a situation where the state of the population is continuously changing without repeating itself during the time when we're asking the animal to keep track of how much time has elapsed. But if the animal were actually using this information to guide their choices, then if this pattern evolved more quickly or more slowly, we should see systematic changes in the patterns of judgments exhibited by the animal. So to look at that, we focused on simultaneously recorded populations of neurons, just 10 to 20 some odd neurons in this case. And we can kind of schematize their activity during this task in a neural state space, with activity starting down here, and then evolving along some typical trajectory, reproducible trajectory during the delay period. And then what we can do is we can take the state of the neural population, at the offset of a stimulus, say this near boundary short stimulus, schematized like this, and then we can project that activity state onto the typical trajectory. And what this does is it gives us a number in the fraction of the average trajectory, or the typical trajectory traversed, that quantifies how fast on average the population has moved along its typical trajectory. Okay? Now, of course, from trial to trial, that mapping is variable, and so we can leverage that variability to ask the question that we set out to ask, namely, does variability in the speed with which population activity evolves correlate with the animal's judgments? So we can then relate a population state to the probability that the animal makes a long choice. Say we can take sort of average speed trials from the middle tercile of this distribution, faster trials from the top tercile, or slower trials from the bottom tercile. And when we do that, we see this systematic relationship between the tendency of the animal to categorize an interval as long and this metric of population speed taken from single trials. If we do that over all stimuli, we see those differences are systematic. They're there for more difficult stimuli. They're not for the easy stimuli at the ends, consistent with this speed kind of metric correlating with something related to the decision variable reflecting elapsed time. Okay, so this is a nice correlation. We've done a lot of other analyses in these papers that you can see down in the bottom. But of course, correlation doesn't imply causation. So what you'd really like to do to test whether these observed correlations reflect the fact that striatal populations are exerting control over the animal's choices is you would like to take experimental control over just this feature of the population response, how fast or slow it evolves while leaving the general structure intact. Turns out that's kind of a hard thing to do even with fancy optogenetic techniques, so we turn to an older school technique, which is temperature. And that's because in a number of different systems, it's been shown that by altering the temperature of neural tissue, you can change the temporal evolution of activity while leaving its general structure intact. And this is an example from invertebrates from Eve Marder's lab in the crab stomatogastric system. These are 3 neurons in the CPG for the pyloric rhythm. And you can see they fire with a particular pattern at 7 degrees Celsius. If you raise the temperature to 11 or 19, you can see that pattern speeds up. But then if you normalize by the phase, you see that all the phase relationships are constant across these different temperatures. So the speed is changing, but the structure is intact. Many of you are also probably aware of this beautiful work from Michael Long when he was in Michael Fee's lab, where they set out to test whether the kind of precise time-like firing of neurons in vocal area HVC were involved in controlling the kind of temporal evolution of the bird's song in zebra finches. And so they developed a solid-state heat pump and then systematically warmed or cooled area HVC. And what they observed was that the bird's song contracted or expanded in a dose-dependent manner with temperature in its temporal evolution while leaving other characteristics intact, like its spectral content. Okay, so we took cues from these kinds of studies and worked with our amazing hardware and software platform at the Shimpalimo to develop our own custom thermoelectric device for focal closed-loop control of striatal temperature. So first we wanted to know, what does temperature do to striatal dynamics? And to get a sense of this, we wanted to assess that in the context of a preparation where we could look at the effect of temperature on dynamics absent the potentially confounding influence of ongoing behavior, because we'd already observed that behavior was correlated with this speed metric we can extract from the population. So to do that, we adapted an anesthetized protocol from Pavel Ruedo Orozco at UNM in Urquiza, Mexico, that involved expressing channelrhodopsin in a sensory thalamic complex, the ventral basal complex, inserting an optic fiber over the thalamus, a neuropixels probe into the striatum, and our cooling and heating probe alongside it. And so in this preparation under urethane anesthesia, we could stimulate these thalamic neurons optogenetically, get relatively long-lasting but highly reproducible dynamics in striatal neurons, and then we could vary the temperature to see the impact on them. So what do we see? I'll start by showing you a couple example rasters and spike density functions of individual neurons. This is time on the x-axis, trial number, or really optogenetic stimulation number on the y-axis. And then there's this sort of jagged schematic highlighting how we're varying the temperature of our cooling and heating probe in 3-minute blocks, always going from some control, physiological temperature, to first we go cool, then control, colder, control, warm. So we've got 2 cold temperatures and 1 warm. If we look at the initial response, we can see the immediate a direct firing of the neurons in response to the optogenetic stimulation. But if we look at the kind of longer timescale relaxation dynamics of the system, okay, this is long after the optogenetic stimulation is turned off, you see we're able to just push around, stretching and contracting the response of this neuron. And you can see that in the spike density functions above. This is one neuron. I have no idea why my computer does that. I am. There we go. So that's one neuron, that's another neuron. Here's the aggregate data expressed in terms of the percent dilation of the spike density function compared to spike density function computed from a subset of the control temperature trials. And so you can see in the aggregate, we have this really nice kind of monotonic dose-dependent effect on temporal scaling by temperature. Great, so it looks like we have this tool that we set out to acquire to be able to stretch and contract the speed of population responses. But before I move on and show you the effect on behavior, this wasn't the only effect that temperature had on firing. Temperature had a qualitatively distinct effect on just baseline firing of neurons. So both warming and cooling the striatum lowered baseline firing rate. Right? So we have a non-monotonic effect of temperature on firing rate, a monotonic effect on temporal scaling. And that's going to end up being important later. Just put a pin in the firing rate result for now. And let's go ask what happens to animals' behavior when we vary temperature in the striatum. So if we look at the probability that the animal makes a long choice, it's a function of interval duration during control blocks, looks like that, warm blocks, cool blocks, cold blocks. This is aggregate data over 6 animals. If we look at each animal separately by plotting the threshold of the fit psychometric function, we see that it's a really robust effect that's there in all animals. So very clear bidirectional dose-dependent effect of temperature on the animal psychometric curves as predicted by the earlier correlative study. So great, we're done, right? This is like your classic systems neuroscience experiment. You go in and you identify some brain structure involved in a behavior, you record neural activity there, you find a correlate of that behavior, okay, then you go in and you perturb activity there and you see that you can push behavior around in the way that the correlation suggested, and you're done, right? And this is actually, I think, this is a really striking result. And I think it highlights a very general principle that's popping up all over the brain, which is generally that the kind of rich dynamics possessed by neural populations provide a really good temporal basis for a range of different computations, okay? And here we think they're involved in this process of policy learning where temporal information is being mapped to one of 2 lateralized actions, but how those dynamics are used in other areas depends on where they're situated. All right, but we realized there was actually an additional opportunity with these data to ask an even more general question about the specific behavioral control functions of the basal ganglia. As focal manipulation of temperature has proven very useful for linking neural dynamics in a particular region to specific behavioral processes, as pointed out in this nice review by Mike Long and colleagues. Now, in the basal ganglia field, there's a bit of a debate going on regarding whether the moment-by-moment evolution of activity there reflects online control of movement execution, in which case perhaps we might also observe temperature-dependent effects on movement trajectories. And this could perhaps explain the effect on decisions that we see as resulting from some kind of embodied strategy, right? The animal develops some dance, basal ganglia is involved in controlling that dance, they look to where they are in that dance at the time of the second tone, and they make their choices accordingly. Alternatively, perhaps striatal dynamics might reflect something like the time-varying drive to select or suppress discrete actions that's read out by some downstream structure to help mediate these more punctate behavioral transitions, such as the kind of decision boundary where we think the animal is switching from planning a movement to the left to planning a movement to the right. Okay, so we wondered if we were to look at the animal's kind of continuous movements, would we see kind of monotonic temperature-dependent effects on their movement trajectories? And we did not. For example, temperature didn't affect the paths in the space of movements, in space of the movements that rats use to report their judgments. So if we track their head as they move to these lateralized ports, you can see example single trials from control blocks here, and then the superimposed averages across the different temperatures. They don't look obviously different. If we splay that out in time, okay, no obvious differences in the time profile. If you look at the envelope of velocity, again, the general shape of that function looks similar across the different temperatures. However, if you squint, you might notice it looks like there may be an effect on just the overall velocity of these movements. And in fact, if you plot average implant speed, which is what's being tracked, as we manipulate temperature, you do see a more modest but significant effect on movement velocity. But it's non-monotonic as a function of temperature. So it's qualitatively distinct from the effect that we see on the animal's decisions. right? Which we interpret this result as reflecting the well-known role of basal ganglia neurons in the control of movement vigor, okay? All right, so backing out, what do we think this means for the general question of the contribution of basal ganglia circuits to behavior? Well, these kinds of measures over here, we would see them as reflecting something like online continuous control of movement and execution, right? And these results here suggest that, you know, the basal ganglia is relatively unconcerned with that, save for perhaps a kind of relatively low-dimensional gain-like modulation of movement vigor, right? In contrast, the moment-by-moment, changing the moment-by-moment evolution of population activity in the striatum does seem to have profound effects on when in time these kind of discrete transitions between two behavioral possibilities are placed, okay? We might view this as a kind of more discrete kind of control, where the controller's action space is fundamentally more kind of categorical and discretized. It turns out this is there's a big difference in control strategies you would use, say in engineering, for more discrete versus more continuous control problems. And so this led to us beginning to speculate a bit about a link between algorithms and brain systems. Okay? So all reinforcement learning algorithms have to learn a policy, this program for behavioral control that maps states of the world onto actions. But there are different ways to do this. For value-based reinforcement learning, the kinds of algorithms that have dominated much of the conversation around neural RL, the policy is derived from intermediary representations of value that encode your expectation about the reward you'll get from taking a particular action. Take Q-learning, for example. So you come into some state of the world, S of t here, and you've got these 6 actions on offer, right? You might choose action 4 because it's the most valuable. It has the highest quality, right? That's where Q comes from. Well, let's say you choose action 4, but the outcome is worse than you expected. Well, then you should get a negative reward prediction error, which should lead you to decrement the value of action 4. And so the next time you come into this state, you might choose action 6 because it's the most valuable now. And you'll go on like this, jumping around your actions, gaining experience in this environment until you settle on a decent policy. And that's okay because you've only got 6 actions here, so you can afford to kind of jump around a little bit. It's not going to be too sample inefficient, okay? But imagine if you had a much higher dimensional action space, where actions were specified by continuous variables instead of in this tabular way. You can imagine that you'd be jumping all over the place, and that might lead to strange and unstable behavior. Think about learning to play a game like tennis. You don't do wildly different swings every single time you try and strike the ball. You gradually adjust the features of your movement until you get it right. Lucky for us, the brain has other adaptive control systems, and reinforcement learning offers other classes of algorithms that deal better with continuous control. For example, in direct policy gradient methods, a policy is parameterized by these set of parameters theta here, and the policy is learned directly in this parameter space by trying to climb to the highest point in the landscape of expected reward. Instead of learning being driven by a scalar error signal, like say a reward prediction error, as in Q-learning, learning is driven by a higher dimensional error of sorts, the gradient of expected reward with respect to the policy parameters. I would point out that this is closer to the kind of higher dimensional error that comes with more supervised learning that's been classically associated with the cerebellum. And cerebellar researchers have increasingly identified reward and reward error signals in the cerebellum. So maybe it's not crazy to start trying to integrate the classic internal forward model ideas with a more directly control-adapted framework such as RL. Now, under this scheme, learning proceeds smoothly because you're moving locally in parameter space. And continuous actions can be accommodated more easily because of the parameterization. Because of the parameterization. So maybe evolution has kind of hit upon the fact that different algorithms are kind of suitable for these different classes of control problems. And that by having a hybrid system for learning control, you get the best of both worlds. This one over here in the basal ganglia might allow you to kind of avoid local maxima or minima, depending on how you frame the problem, that you might end up in using this gradient approach, whereas this other one allows you to converge smoothly to an optimal policy if you're in the right neighborhood, okay? Now, we would assert that an interesting candidate for this latter function when these continuous control parameters are subject to learning is the cerebellum. Okay, so we've begun to examine behavior that more explicitly requires continuous control and coordination. And this is work that was started as a collaboration with Megan Carey and John Krakauer, with contributions from Coralie Arendt. The main people leading it are Teresa Duarte and Simone Zamora, as I mentioned. And this is a head-fixed reaching task where mice are reaching for droplets of water. We can characterize the behavior at high temporal resolution using high-speed videography and markerless tracking algorithms. And one of the first things we did is just ask, well, what happens when we manipulate the temperature of the cerebellum? And I'm just going to show you 3 example trials here. One trial from a control block. Okay, one trial from a warm block. The animal actually misses it. And one trial from a cool block, where you see they make this kind of hypermetric overreach. Okay? And if we look at a larger number of reaches from this animal, okay, you can see what you saw in those individual trials, namely that warming seems to produce this sort of less eccentric, hypermetric movement trajectory, whereas cooling produces this really eccentric, hypermetric movement trajectory. And so let's think about this for a moment. If you suppose that the moment-by-moment evolution of population activity in the cerebellum encodes kind of moment-by-moment commands that are going to guide like online control, and then you take the time base for those commands and you contract them, right? Let's say those commands are something like go forward, forward, left, left, back, back. Well, now you're going to start turning left sooner, right? And so this is sort of potentially consistent with thinking about the output of the cerebellar circuit being something like continuous control commands for online control. Okay, so that was one mouse. This is another mouse in a different set of experiments where we applied temperature manipulations both to the cerebellum and to the striatum. And you can see the same kind of thing. So now we're looking at the paw velocity as a function of time aligned on reach endpoint. Okay, you can see that the temporal profile of the velocity is kind of squished or contracted in a dose-dependent, temperature-dependent way, whereas we don't see that when we do that manipulation in the striatum. Conversely, we added in a kind of discrete control component into this task, namely that animals have to pull on a manipulandum in order to initiate the trial and elicit the reward, right? But trials weren't available to be initiated until 5 seconds after the last reward. And as anyone who trains animals knows, if you put fixed time intervals into tasks, animals will predict them, right? And so we hypothesized that animals would try and time their trial initiations to this 5-second interval, and that when we change the temperature of the striatum, we should be assaying the same kind of discrete transition-like control function that we were assaying when we were looking at the decisions in the rats. And indeed, we found that to be the case. So we saw the same kind of bidirectional dose-dependent effects on, in this case, timing of movement initiation to initiate the trial when we manipulated the temperature of the striatum, and we saw no hint of such an effect when we did the same in the cerebellum. Okay, so these 2 slides, they kind of represent this striking double dissociation in terms of the effect of temperature on different aspects of motor control. And we're really interested in trying to dig deeper and understand at a more formal level what this means. Okay? We're also doing a ton of other different kinds of experiments. By we, I actually mean Teresa, Simone, and Ugo. Those include large-scale simultaneously recordings from cerebellar cortex, deep cerebellar nuclei, brainstem, sensory motor cortex, sensory motor striatum. And unfortunately, they presented their poster last night. I mean, it's not unfortunate they presented. They did a great job. But they've already presented it, so you won't be able to go see it later. But if you see them, you should go ask them to tell you a little bit about some of the early results they're seeing from the analysis of this data. Generally speaking, I'll tell you, so far they seem broadly aligned with this idea that there's this division of labor between discrete and continuous control across the basal ganglia and the cerebellum. Maybe just in the last piece of data I'll show you, this is actually work done in collaboration with Megan Carey's lab by Coralie Arendt together with Teresa. What Coralie did is she induced genetically cell type specific lesions of particular cells in either the basal ganglia or the cerebellum. Specifically, she used CASP3 to produce a basically complete lesion of indirect pathway medium spiny neurons in one set of animals, and diphtheria toxin receptors to produce a basically complete lesion of Purkinje cells in the cerebellum. All right, and I'm just going to show you 3 brief video clips of mice performing a freely moving version of this reaching task, where they have to reach through a slot in the wall in order to get the water droplet. So first, if you look at control animals, they go up to the slot, they rear up, they reach through it, they get the water droplet, and they bring it back out of the slot to consume it. If you look at the animals that are missing their indirect pathway medium spiny neurons, okay, they come in, and they basically start doing kind of all the actions all at once, okay? So they're licking the entire time. And again, control animals almost never lick during the reach, right? But these indirect pathway animals, they're basically, it's like they have all of the right actions loaded up, but they release them all at once and can't properly sequence them. As if the indirect pathway is normally involved in kind of keeping particular behaviors in the set in check until you're at the appropriate point in the sequence to execute them. Now, Purkinje cell lesion animals, they're badly ataxic, okay? So they can't actually achieve any reaches. These are tough videos to watch, actually. But what you'll notice is that this guy's trying to do the right thing. I'll just try and play that again. He's trying to do the right thing, okay? So he goes over to the slot, and he tries to rear up. So it's like he initiates the right thing, but then he's completely incapable of controlling it, right? So again, we interpret these kinds of observations as, broadly speaking, being aligned with this hypothesis that there's this distribution of function across discrete and continuous control. Okay, so just to wrap up, we think these data suggest that behavior might be viewed as sort of resulting from the concatenation of discrete selection-like acts to determine what to do and when, interspersed with bouts of continuous online control. And our working hypothesis, based on, in part, the data I showed you today, is that we might view the cerebellum as a system for learning policies that deal with these bouts of online control, while the basal ganglia might learn policies for selecting amongst some discretized set of actions, perhaps served up by other cortex or brainstem circuits, such as those that Sylvia Arber's lab has described so beautifully. And perhaps they achieve their respective functions by implementing different classes of what might be considered RL algorithms, with cerebellum learning policies directly using, say, gradient-based policy learning, and the basal ganglia resting on something akin to purely value-based methods. Okay, so the division of labor between the brain systems that I'm focusing on here today, we sometimes refer to as the hierarchy of learning and control. These are different functions without an obvious ordering between them. They're both pretty fundamental and important. But in focusing on that aspect of nervous system function, namely the kind of modularity of brain systems and the distribution of labor across them, I've ignored another important and prominent feature of all major learning systems. And that is that they're organized hierarchically. Both the basal ganglia and the cerebellum operate on a hierarchy of representations, for example, those you find across the cortical hierarchy, that vary in degree of spatial temporal abstraction. So if this concatenation of discrete action selection and continuous or discrete selection and continuous online control functions is an appropriate way of thinking about what these systems do in general, presumably it's occurring in parallel across this full stack of hierarchically organized information, increasing in timescale and abstraction as you go up the hierarchy. And while I've been edging towards motor control mostly in this talk, at the higher levels, we might consider whether these kinds of processes could form the building blocks for more cognitive processes, and whether all levels jointly might be a good substrate for the overall hierarchical organization of behavior. And so perhaps a truer way of talking about the functions of the two learning systems is not to talk about action selection or online movement coordination, but rather more generally selection and placement of switches in dynamics by the basal ganglia and of continuously controlling those dynamics by the cerebellum, which when you bring in hierarchy is kind of a layered switching dynamical system view of brain function. If you want to hear more about how we think about hierarchical control, please go see Renato Souza's poster tomorrow afternoon. Okay, so with that final 10 seconds, we've come to believe that this distinction between discrete and continuous action spaces is really fundamental. I got my start in experimental neuroscience at Tufts University, working with a guy named Barry Trimmer on these guys. These are the tobacco hornworm caterpillars, Manduca sexta. And I was studying central pattern generators for these two classes of behaviors that these guys exhibit, either forward crawling or casting. Okay? And I've been thinking about those experiences a lot lately, and specifically how once an organism has access to multiple central pattern generators, it's faced with these same two problems I've been talking about. Namely, which CPG do I select and when? And once I've done so, how do I continuously control its output? So this too, like the need to absorb temporal dynamics into control functions, we don't think of as being limited to learned control, but goes all the way potentially down to the lowest level of biological control functions. So with that, I'd like to thank the people who did the work and provided a lot of the thinking, all of the people at the Champalimaud Foundation who make doing science there so enjoyable and possible, the funders, and you for your attention.
Thank you, Joe. It was great. We have time for a few questions. So, and we want to encourage young trainees to, also ask questions. Don't feel. I know it can be, feel quite stressful, but go for it. Okay.
Me?
Yeah.
Hey, Joe. That was a great talk. When you think about the hierarchical structure, do you see the continuous control at hierarchies above the discrete control as well as the other way around? So is it like, can you imagine where you have continuous abstract space and then discrete selection below that, and then continuous selection at the level of individual motor behaviors, and that might be a structure that goes all the way up?
Well, I think I see it the way I kind of drew that, that schematic, right? The joints between like kind of fixed dynamics are going to be at different points depending on what layer of the hierarchy you're in, right? So this would be a kind of nice potential substrate for like hierarchical control where you choose an action that's kind of temporally abstracted, right? And that's going to be in place for a long time. And then that defines some sub-goals, right? And then within each of those sub-goals, you're executing on particular dynamics and you have to switch between them, and so on and so forth as you go down to sub-goals of sub-goals and so on and so forth. Does that make sense?
Yeah, it makes sense. I just think it's an interesting, provocative idea to imagine that you might have these. So the way you framed it is a simple switching linear dynamic, switching dynamical
system. Doesn't have to be
system,
linear, yeah.
Yeah, it doesn't have to be linear. Mm-hmm. Switching dynamical system, which I think is a simple way to conceptualize it. And I was wondering if you could imagine an experimental framework where you try to look for continuous layers in more abstract spaces. We can talk about it.
Yeah, we can talk about it. Yeah.
Yep. Thank you for your talk. So regarding the striatal dopamine specifically, would you interpret your results as dopamine manipulating the integration time scale, or managing the learning and implementation of the order of the sequences? Because maybe it's trying to order the sequences, the behavioral sequences properly, or maybe it's compressing or stretching the division of the behavioral syllables. Maybe, what do you think about this?
Yeah, I mean, we've shown in a previous paper that dopaminergic neuromodulation does seem to be able to affect behavior on this task. We've been trying to do results where we can kind of stimulate the dopamine and look at the effect on dynamics. Those are tough experiments, so I don't have an answer for you there. But generally speaking, the way we're thinking about the effect that temperature is having is that there's a larger kind of reentrant circuit, even though Sean and John Murray have shown that you can get the fully inhibitory circuit in a model of the striatum to produce time-varying kind of dynamics. You have to keep providing drive to it. We think actually that's probably due to kind of longer range reentrant circuits that go through frontocortical areas, in part because other people have done temperature manipulations in medial wall frontocortical areas and found effects on timing. So we think we're really kind of directly affecting population dynamics by kind of intervening at one point in a larger kind of recurrent node. But yeah.
Thank you.
There's some thought that the striatum can have differential effects on behavior for naturalistic behaviors compared to more detailed kinematics. Have you thought at all about that? And yeah, have you run any experiments with like more detailed motor sequences?
I don't know. Do you work with Benza?
I used to.
Yeah. Yeah, I mean, I don't think that these kind of lateralized head poking movements are particularly ethological, and neither are head fixed reaching for water droplets. We see that animals have to learn how to do them. Their performance gets a lot better over time. So I think that we now have evidence from two different behaviors, both of which I would argue are kind of learned and not like innate behaviors, that we're not getting effects on movement kinematics by manipulating the moment-by-moment temporal evolution of striatal activity.
Okay, thanks.
Yeah.
Really nice talk. I'm curious about the learning mechanisms in the cerebellum. So you alluded to them being potentially policy gradient style algorithms, and I'm wondering why specifically those. You could also think about other model-based algorithms which would also result in continuous. Okay.
Totally. Yeah. So I mean, one way to bring in the sort of more classic views of forward model learning by cerebellar circuits is to say, well, it's a model-based reinforcement learning, policy learning system, and it's just operating on continuous action spaces via non-purely, value-based methods. Yeah, so that's definitely a possibility, and I think that's a nice path to pursue in terms of reconciling the sort of historical theory around cerebellar circuits and kind of what we're saying.
Thank you.
Hi, I really appreciate the sort of dichotomy between selection and specification, or sort of high-level and low-level information. But I'm wondering your thoughts on when they're integrated, sort of on the way out of the brain. Are we thinking that single cells do one, or are they mixtures? Like, sort of, at what point are these policies combined, or how might they be combined?
Yeah, so I mean, I sort of alluded to it when I was talking about muscle spindles, just sort of how little we really understand about the kind of lower levels of sensory motor control. So I think I'm sort of content to remain agnostic in the absence of really understanding what is the control space that gets offered up, from brainstem and spinal circuits. And I think there's a ton of sophistication there. And I think you're already seeing signatures that you're not. A lot of, like, say, spinal interneuronal circuits, they're offering up kind of movement primitives that are already in some sense kind of discretized. They're not really in muscle space anymore. So anyway, I'm agnostic at this point, but I think, the idea would be there's some output in terms of pattern output from the basal ganglia. Maybe it's going down to the brainstem, maybe it's going up into thalamus, right? And that's sort of picking out particular circuits or dynamical regimes, and then you maybe have some learned kind of forcing functions that operate on maybe some different higher dimensional space in those downstream circuits.
We have time for one more question.
Thank you for the talk. Do you think that time estimation happens internally in the striatum, or does it come from other regions like the hippocampus?
No, I think it's you're getting kind of estimation of kind of longer timescales by essentially taking into account dynamics that are driven by mechanisms that are spread over kind of larger circuits. Like I said, that our hypothesis, they include kind of frontal cortical areas. So we actually know from, I didn't mention this, but in the rat experiments with temperature, Tiago and Philippe and Margarida put the probe in overlying motor cortex, okay? And we didn't see effects on the animal's choices when we did that. And that's why, I'm saying I think it's probably higher cortical brain structures, not kind of low-level sensory motor.
Thank you. Okay, so the next speaker is Margaret DeMaegd, and she's coming from NYU. She's going to tell us about multi-regional circuit mechanisms of belief updating.
Oh, try this instead. Hey, awesome. All right, so my name is Margaret DeMaegd, and I'm in the Constantinople lab, and I'm really excited to share with you this work I've been doing studying a multi-region circuit for belief updating. We've just submitted this, so you can find it soon on bioRxiv. So inference is a core cognitive computation, and there are many reasons why animals might use inference to guide their behaviors. For example, if the sensory inputs are ambiguous, or you want to understand and respond to the internal states of others, or to the changing states within the environment. And our lab seeks to understand the neural circuits that support this last use of inference, and I have identified a cortical striatal circuit that plays an important role for updating and maintaining beliefs about changes in the world. So to do this, we developed a temporal wagering task in which rats reveal their subjective value of reward offers by how long they're willing to wait for them. Briefly, this is a self-paced task where rats must nose poke into a center port and hold there while a tone conveys the reward offer. Then a side light will turn on, which will indicate which port is going to be rewarded, and rats can choose to either wait for an uncued, unpredictable delay for the reward to arrive, or at any point in time, opt out by poking into the opposite side port. Now, on a subset of trials, the reward is withheld, so the rat must choose this opt-out option. We find that rats are sensitive to the reward offers, such that they wait longer to opt out for larger reward offers, as you might expect. Additionally, there's a hidden block structure, in which the reward distribution alternates between 3 states, a low, a mixed, and a high state. And the transitions between these 3 states are uncued, so the rat must infer which block it's in, given the reward history it's just experienced. So one indication we have that rats are sensitive to the changes in the reward state is that they modulate how long they're willing to wait for the rewards, depending on the reward state. So here I'll point you to the 20 microliter rewards, which are present in all 3 blocks. And what we find is that rats are willing to wait a lot longer for that 20 microliter reward when it's the best option in a low block in blue than when it's the worst option in a high block in red. Furthermore, previous lab members developed an inferential behavioral model inspired by foraging theory. This model uses Bayes' rule to infer the most likely block, and then implements a fixed block-specific wait time threshold determined by the opportunity cost of each block. This model shows similar modulations in wait times as what we observed for the rats. Now, I understand this is just a single example, but this model also captures other qualitative and quantitative aspects of the animal's behavior that I don't have time to show you today, but you can check out the many other publications from our lab that go into more detail. For this presentation, however, I'm going to turn your attention to the orbital frontal cortex. So the OC has been implicated for inferring values in partially observable environmental states by a variety of labs, including the Schoenbaum, Mayman, and Kepish labs. But specifically within our lab, a recent grad student, Shannon Schierick, showed that inhibiting the OFC using a GABA-A agonist musmol caused rats to be less sensitive to the changes in the reward state, such that they reduce how much they modulate their wait times. Again, I'll orient you to these 20 microliter rewards, where in the inactivation state, in the lighter colors here, you can see that they're far more similar to one another. Additionally, though, we find that rats are also slower to detect transitions from one state into another, which is what I'm showing you here, from a mixed into a low in blue and a mixed into a high block in red. And so the inactivation, again, are these lighter colors, and while eventually they get to the same point that the control animals do, they take much longer to do so, and they need far more evidence to update their beliefs. So I was really interested in this, and I sought to understand how projections out of the OFC helped to guide the animal's behavior. So to do this, I optogenetically activated OFC projection neurons while the animal is collecting its water reward. And the OFC is known to project to the striatum, so I virally targeted those projection neurons using channel rhodopsin. And what I find is that optogenetically stimulating these OFC to striatal projection neurons reduces wait times, such that we see the greatest effect in the low blocks, an intermediate effect in the mixed block, and a little influence in the high blocks. And so before I dig deeper into sort of the cognitive basis for this, I want to express kind of how amazing this effect is. So I'm stimulating at reward for 500 milliseconds, but I can only measure how long the rat is willing to wait on trials where the animal chooses to opt out. And so these are, by definition, two separate trials. This indicates that the modulation has these long-lasting effects that carry across trials. Okay, but we want to know the cognitive basis for these changes in the wait time. So we hypothesize that the cortical striatal activation is biasing the rats to believe that they're in a high block, as this would account for the strong changes that we see in the low block and the weak effects that we see in a high block, where they already believe they're in a high block and so are unlikely to change their behavior further. To test this hypothesis, we instantiated a bias into our behavioral model, such that the probability of believing that it was in a high block was increased, and the probability of believing it was in a low or a mixed block was reduced. We just asked whether this resulted in the same changes in wait time as we had observed in the animals. We found that this bias did reduce the wait times in the model, with the strongest effect for low blocks, which you see here in the lighter colors, and matches the animal's behavior that I had just shown you previously. But what's really cool about this model is that we can use it to make predictions about the animal's behavior that I hadn't already assessed. For example, we found that the model was really slow to detect transitions specifically from a mixed into a low block, which is what I'm showing you here. And so indeed, when we went back to the animal behavior, we found a similar result. The rats needed more evidence to detect this transition from a mixed block into a low block. What this together means is that the optogenetic stimulation is biasing the animals prior towards a high block. So we think of these two experiments as consistent with one another. The inactivation of the OFC makes the beliefs resistant to updating, whereas projection-specific activation biases the beliefs to update directionally towards a high block. And that's great, but I wasn't satisfied, and so I wanted to know what information is being sent to these downstream circuits. So I performed electrophysiological recordings from the OFC using neuropixels probes and identified these cortical striatal projection neurons optogenetically by tagging them with channel rhodopsin and looking for evidence of action potential collisions between spontaneous orthodromic spikes and LED-elicited antidromic spikes. Using this technique, I recorded from 60 projection neurons and found that they positively encode reward. As you can see, they have higher firing rates for the larger reward volumes, which are in the warmer colors in this example neuron. But what we find really exciting is that if we average the response across the full population, the firing rate still varies with the reward, but 40 and 80 in red and orange, and 5 and 10 in blue and purple are overlapping with one another. And this is really important to us because these pairs of volumes provide equal evidence for the underlying state. That is, 40 and 80 provide equal evidence that the rat is in a high block, as do 5 and 10 provide evidence that they're in a low block. So this activity looks less like a graded representation of volumes, but instead a categorical evidence of the reward states. In other words, this looks like the likelihood of a given reward that the rat is experiencing, given that they're in a high block, which is amazing. And so we thought to take this a step further and ask whether we could decode the most likely block from these neurons' activity. We found that not only could we decode that most likely block greater than chance, but we could also decode the second most likely block as well. And they do so better than the full untagged OFC population, which includes many different types of projection neurons, including those that are projecting to the striatum. This is really interesting because there are 3 blocks, and their probabilities must sum to 1, representing the first and second most likely block actually provides sufficient information to represent the full probability distribution. So this work provides novel insight into how specific projection neuron classes, explicitly represent probability distributions over discrete categorical variables. Finally, because Shannon Sherrick had previously shown neural correlates of inferred states in the OFC, we wanted to ask, does stimulating these OFC to striatal projection neurons disrupt representations of inferred states in the OFC dynamics themselves? So to do this, I recorded from OFC population dynamics while optogenetically stimulating these striatal projection neurons during reward, as I've just shown you. And then we used a dimensionality reduction method called DMIX-PCA to identify latent factors reflecting the encoding of block, reward, and time. And we applied the same weight matrix that we trained while the LED was off to the same neurons that we recorded while the LED was turned on, to project activity onto the same demixed principal components. And then we asked the big question of whether or not these OFC striatal projection neurons are modulating OFC population representations of the block. Okay, so here I'm showing you the top block demixed principal component, where the mixed block activity is represented by the solid lines, and the high block is represented by the dashed lines. So prior to projection neuron modulation, there's a clear separation between the two of them. But this is diminished after optogenetic stimulation. This indicates that the OOC block encoding is disrupted by activation of these projection-specific neurons. Now, this is just one example, but the effect is also true across sessions if we look at the difference in encoding between the two blocks. However, we find that the reward encoding in the OFC dynamics is unaffected. And so what this tells us is that these projections are critical for updating the inferred states, but not the rewards. Finally, we sort of wanted to know how these projection-specific neurons are modulating the OFC dynamics. And to sort of reorient you, we thought of two broad possibilities. So I'm stimulating at the axon terminals, meaning that we're either modulating striatal activity and driving the OFC through these long cortical basal ganglia thalamic loops, or our stimulation is driving these antidromic action potentials to modulate the OFC directly. This is a little bit tough, but we can do it. So we measured the number of extra LED elicited action potentials in the OFC, in these projection neurons specifically at the time of reward. And on average, what we find is that there's less than one extra spike that's detected at these projection neurons in the somata. And we think this is likely because these neurons are already active at reward, so there's more collisions. But we also have other evidence to support that there is network-level inhibition that limits the peak firing rates that I don't have time to show you today. So this suggests that this direct influence on the stimulation, or on the OFC dynamics through stimulation is rather unlikely, and instead we're sort of falling back on our alternative hypothesis is that we're acting through these longer cortical basal ganglia thalamic loops. Okay, so to put it all together, the OFC supports belief updating. These OFC striatal projection neurons update the beliefs towards high blocks, and they do so by encoding evidence for the high blocks with higher firing rates. They also are impacting OFC representations of the inferred state, and they're likely doing this through a cortical basal ganglia thalamic loop. With that, I'd like to thank the organizers for this opportunity, our funding sources, and then especially the Constantinople and Savin Lab for their support, especially the people that are listed here whose work directly contributes to this project. Thank you.
Thank you. We've got time for questions. Well, maybe I can get started. So I was trying to relate your talk with the previous talk from Joe. Do you think that some of the effects you see here could be a consequence of loss of discrete control of actions? Or, yeah.
Ooh.
That is probably a tough question.
We haven't really thought about it. So I have video that I've looked at, and we don't really see major changes in their behavior, but I haven't really quantified it really intensely. So I'm tempted to put it more on the beliefs, but yeah, I don't know for sure.
Thank you.
sort of related to the, great talk, by the way.
Yeah.
That was sort of related to the question I was going to ask, but I was wondering whether, I wouldn't necessarily expect to see effects on actions per se in like sensory motor circuits, but it was noteworthy that you specifically said you think you're looking at probability distributions across kind of like discrete task states. So do you think, now sort of a twist on Rui's question, could you think about that as sort of, the basal ganglia being involved with orbital-frontal circuits and kind of, selecting between beliefs about the current state of the environment across this discrete probability distribution?
I think that would make sense.
Okay, cool. Good. It's consistent with my worldview.
Yeah.
Thanks.
Okay. Yep.
Great talk, thank you. I'm wondering, do you have an hypothesis as to why these like OFC to striatum connections would encode the probability of being a high block specifically and not a whole block, a low block or a medium block?
Yeah. So we see some variability. So I've sort of glossed over it. There's some heterogeneity in the neurons themselves. That there are a subset of population of these projection neurons that tend to encode the low block with higher firing rate. As to like exactly why the higher firing rate for the high blocks, I don't know totally. It's bothered us as well.
Thank you.
Yeah.
Okay, thank you.
Yeah.
Okay, so the next talk's by Tomas Aquino, and he's going to tell us, it's coming from Columbia University, and it's going to tell us about human medial-frontal neurons integrate language and vision to structure decision making.
Okay. Okay. Hey everyone, my name is Tomas Aquino. I'm really happy to be here. I'm a postdoc at Columbia University at the Theta Lab. So today I'm going to share our new work on how medial-frontal neurons encode the semantic contents of instructions during decision making, and how we use artificial neural networks to model the process. So an open question in cognitive neuroscience is, how exactly do verbal instructions interact with sensory evidence within these neural populations to guide flexible behavior? For example, what needs to happen in human neural populations for us to be able to follow GPS instructions accurately? While there's extensive non-human literature on this topic, flexible decision making, studying natural language instructions affords a number of new unique opportunities because humans can follow instructions that we have not seen before, and we can quickly adapt to new and even opposite semantic contexts. So we decided to tackle this open question like this. Given a pair of linguistic and sensory inputs, how are human neurons integrating these factors to then go and compute goal-directed decision-making factors? So to answer this, we obtain neurons from intracranial recordings in sensory and decision brain areas from epilepsy patients. So this is done in collaboration with the Rudeshauser lab at the Cedars-Sinai Medical Center. With these, we can also ask if there's some sort of hierarchy in encoding across sensory and decision neurons. Our patients performed a task that mixed visual stimuli with verbal instructions. From their perspective, the task is quite simple. They are given one linguistic instruction every trial, such as choose the oldest car, and 2 visual stimuli, such as 2 photos of cars. Then they have to report which of the 2 stimuli is the correct choice. I should mention here, just for the interest of time, there's a total of 192 trials with 4 possible categories of photos and 4 types of comparisons that we're asking patients to do in each trial. Additionally, for half of the trials, we inverted the intended semantic meaning of the instruction by giving patients a negative instruction. So for example, we would ask patients, do not choose the oldest car in trials like that. Our end goal here is to then go and provide a computational model for how this sensory linguistic integration is performed. So therefore, in collaboration with the Puget lab from the University of Geneva, we developed a visual language model that can perform exactly the same task as patients with the same inputs. So this VLM receives both images as sensory inputs to a CLIP visual model, and the language instruction as an input to the language model as BERT. These modules then go and produce their respective embeddings, which we integrate into a recurrent neural network. In the end, this network produces a binary decision for each trial. Now, I'm going to show some behavior. Here's how the model performed on the task across our task conditions. So this is accuracy on the y-axis. So the takeaway that I want to highlight here is that the model can perform the task pretty well, above 80% in all conditions. And if we compare it to humans, humans can also do the task very well across the board. Maybe the accuracy is just slightly lower than the models, but they're doing the task pretty well as well. Okay, so speaking of our patients, let's get into brain recordings in more detail. We had a total of 11 epilepsy patients here who were implanted with Benke-Fried electrodes during epilepsy monitoring prior to their surgery at Cedars-Sinai. With these microwires, we obtained 834 neurons across these areas, including ventral temporal cortex and pre-supplementary motor area. So what can we do with this data? One thing that's very, very important here, just to situate ourselves, everything that I'm going to show you from here on out is just neural activity from stimulus presentation when the pictures are on the screen. We did this to focus at this stage of information processing where that's not directly related to the motor aspect of pressing a button. We got spike counts for each neuron during stimulus presentation and counted how many neurons are significantly correlated with sensory or goal-related factors in the task. So here the percentage of significant neurons is on the y-axis. For example, did firing of neurons correlate with the visual category of a stimulus? So we do find widespread correlations with this factor, most prominently in ventral temporal cortex. And now we can also look at task goals and things that are presented via instructions. So for example, we see that neurons in pre-supplementary motor area correlate with several factors, and I'm listing only a few of them here for time, but we see that it correlates with which comparison patients are being asked to do, and also with the framing of the task, that is, if you're being told to do something or do not do something. But interestingly, we can also look for correlation with choice. That is, we can actually think about the abstract meaning of the choice of each trial, not pressing buttons, but is the stimulus that I'm choosing the first one in the sequence or the second one in the sequence? Again, we can see that neurons in cingulate cortex correlate with that variable, and this has nothing to do with pressing a button. This is way before that happens in the trial. Okay, so to get a time-resolved sense of the neural code during stimulus presentation, we're also doing pseudo-population decoding. And just to simplify the population level analysis here, we're collapsing hippocampus and amygdala into MTL, medial temporal lobe, and pre-SMA and cingulate into MFC, which is medial-frontal cortex. So that's the nomenclature I'm going to go with forward. On the y-axis here, we have single trial decoding accuracy. Category has a chance level of 25, and the other variables have a chance level of 50. For example, we see here that VTC and MTL do encode the category of a stimulus visually, makes sense. But additionally, if we look at the split magnitude of the visual features, so if it's a high age or low age stimulus, or high count or low count, high color or low color, we observe that VTC is encoding all of these features, this low level visual features. On the other hand, we decoded variables related to the task goals from medial frontal. We can see that these neurons encode the magnitude of visual features along the instructed target axis. Additionally, the abstract choice in each trial is being encoded by patients even during stimulus presentation. We see that as well in VMPFC a little bit earlier. Now, for us to be able to claim that we have some sort of computational understanding of these cognitive processes, it is really useful to build an artificial system that can perform the same task and then use it to predict the behavior of the neural system. So this mirrors work in visual processing with convolutional neural networks. And luckily for us here, we do have a model that performs exactly the same task as patients. So here's what we did. We take the exact same sequence of trials as patients experience them, and we present them to models that were previously trained on the trial on a separate set of trials. From the model, we can then obtain language embeddings, vision embeddings, and then the integrated RNN activity for each trial. If we call these artificial representations X, we can then use them to predict the firing rates of each neuron Y. If the model is able to predict neural activity out of sample in trials that the predictor hasn't seen yet, then we can claim that we obtain some generalizable insight about how neurons are computing the task. For example, let's say that X are the visual embeddings from CLIP. Can we predict neural activity in left-out trials from that? On the y-axis here, we have neural activity for each trial. In gray, we have the estimated neurons activity from image embeddings. And then in red, we have the spike counts, the real spike counts in those trials. This is the raster plot for that neuron that we found. We can see that it is a category neuron that fires more for images of cars for some reason, right? The model has the ability to pick up on that pattern and then therefore predict the neuron's activity. We can do the same with pre-estimated neurons that track more specialized patterns. So for example, this is a neuron here that fires more when age is a relevant feature as instructed. To summarize our ability in the entire neural population to predict models, we obtained this out-of-sample variance explain metric, R-squared. We compare it to a null distribution just to be able to claim that these neurons are being explained better than chance. And here we can see that 44% of neurons in VTC and 12 in MTL are explained by CLIP's visual embeddings. If we go post hoc and look at each of these significant neurons, what are they doing individually? We can see that most of these neurons that are significant in VTC and MTL are indeed category neurons, and the model is able to pick up on that. If we use the semantic text embeddings from SBERT, we see that 14% of neurons in VTC are also explained by it. Again, mostly category neurons here. And very interestingly, for me at least, the integrated signal that exists in the RNN, that's the decision part of the model, it explains the diversity of activity across all brain areas. The category signal of VTC is still present there, but if we look at MFC, we can see that 37% of neurons in medial-frontal are explained by the RNN. And this includes neurons that do target feature coding, choice neurons, and neurons that do the framing of the task. There's a lot of things here. But here's one really exciting reason for us to be doing this. We're able to predict spikes of other neurons that correlated with variables that we had not previously tested for, that we weren't looking for. So for example, here, just for the interest of time, we found neurons that are coding the task state. That is, am I looking right now at the first or the second neuron in the sequence? We hadn't previously tested for it, and the model pick up on those neurons for us. So here, to summarize everything, we map ways in which sensory and linguistic decision-making can affect activity in neurons in humans. And to summarize our findings here, if we have ventral temporal neurons that correlate with the sensory aspect of the task, but also we have medial frontal neurons that correlate with task goals and choices in a more abstract fashion. And we're also able to provide here some new neuro AI alignment results, especially with our ability to predict neural activity along this sensory decision hierarchy. The work today would not have been possible, of course, without these incredible collaborators at Cedars-Sinai and the University of Geneva. I'm especially grateful to our patients at Cedars and the amazing members of the Natita lab and the Rittishauser labs. So a huge thank you also to our funding sources, and please stay tuned because there's a preprint coming out very, very soon, hopefully. Yeah, thank you.
Thank you. It's time for a few questions. Maybe I can get started.
Yeah.
So you find that RNN performs better at explaining data than the transformers themselves, right?
Say it again?
You find that RNN explains data better than the transformers.
Yeah.
So how should we interpret that? What do you think that means?
Right. I think the RNN is picking up on things that correlate with the task that we might not have been able to handcraft as regressors, right? We also have in the beginning a forward encoding analysis where we handcraft regressors such as task factors and we correlate that, right? So the RNN is probably creating some nonlinear construct that is a function of those task factors that are inscrutable, at least at a first approximation for us. But in my view, it's probably just a nonlinear transformation of task-related factors that happen to be relevant for decision, and then we can predict variance from that.
Okay, thank you. Any questions? I mean, I have another follow-up. Was there a reason why you went, I mean, I can see why you picked an RNN as a way to combine these things, but did you consider using a transformer instead? Or, yeah.
Right. I think it could have worked in many ways just as well. I think one of the reasons we went with an RNN is that it builds up on previous work. So there's like a really solid literature that already tells us what the dynamics should be inside of it. So it's more, it's easier to go inside and do neuroscience of the network, but I think it could have worked just as well. And obviously CLIP has the transformer architecture built into it, so at least a part of this has some transformer-like architecture, right?
That's another question coming up.
Hey, it's super cool to see these models being tested in human cells. And one of the cool things about the original model is that the state representation from the large language model sets off the dynamics of the RNN such that it constrains it into like different initialization constraints. So I just wondered if you'd seen any of those dynamics in the neuron.
Right. We haven't done a lot of dynamics analysis yet. That's a natural next step for us to do. One thing I'm able to say right now is if we go inside the RNN and we look for what kind of representation exists there, we see that it's parsing out, for example, low and high level of the sensory features versus target, stimuli, goal-related things, right? The RNN picks up on that, and those are the factors that correlate with neural activity. But in terms of using that to predict dynamics of neurons, I think this GLM is as far as I got, but yeah, that's a natural next step, yeah.
Okay, thank you.
Thank you.
So now we were supposed to have a talk by Ulises Pereira from the Allen Institute, but because of issues with visas, he couldn't make it. We have uploaded the video, so he has recorded his talk, and we have uploaded it to YouTube. So we encourage you to watch it when you're on your own time. But that's the end of the session. Thank you.